going to talk about our grant program a little bit, and one of the critical knowledge programs that Natalie talked about, genetics, because I love genetics. So, but first about the grant program. And the grant program has been kind of our bread and butter since FPWR started. And in contrast to the programs that Natalie has spoken about, where we're trying to take the lead and move things forward, this is a program in which we ask the scientific community, what are your best ideas? And we have a competitive process by which we select those ideas that are A, scientifically really good, and B, very important to our children. So it's a great way of, we feel like even though we're starting many directed research programs, we want to keep the grant program robust because it's been such a great way to gain insights into PWS, to start clinical research projects, to get things rolling. So the goal of the grant program is really to support those earlier stage projects. So NIH grants are much bigger than FPWR can do, so they're usually 4 or 5 years, $250,000 a year. They're very, very competitive. So one of the goals of our grant program is to give people a head start so they can test out some hypotheses and gain some data so that they can then be really competitive for a bigger grant program, so the start of a new idea. So we want innovative ideas, we want ideas that are a little bit higher risk but have potentially higher reward, and we want ideas that from both new investigators, so young investigators, because if we can suck someone in early in their career, they may stay in PWS for the rest of their scientific career, and that's good for us. And it also gives those established researchers who have a career in PWS, who have a new idea, a way to test out their new ideas. So our grants are selected, as I said, we get scientific reviewers and we get parent advocate reviewers. If you, I know many in the room have been advocate reviewers. If you're interested in that, please talk to a parent who has been, and please send Jessica your email and we'll put you on the list for doing advocate review. And we look for research, obviously, that aligns with the priorities, what all of you have said is important to you. And so we are constantly seeking feedback from the community about what is important to you. So please let us know, because this is your program. So again, we sometimes target, as I said, we didn't feel like we had enough neurobiologists in the field, so we really went out and tried to pull some new neurobiologists in, and we've been successful at that. So this gives us the flexibility to try to bring in the people that we think should be addressing PWS. So this program has been going on since the inception of FPWR, and to date we have invested roughly $9 million in PWS research. Yeah. And I got to tell you, it took us years to get to that first million. So it's really been, we've been on a nice trajectory. 135 projects in in 13 countries. I mean, we're agnostic as to geographic area. We just want to go where the best PWS research is. And so obviously I can't go through all of the things that have come out of that, but the way scientists communicate information and the way they build on each other's work is through scientific publications. And so FPWR has supported work in more than 125 publications in the literature, many of which are in very high-profile journals. So Nature Medicine and Journal of Clinical Investigation, which to me is like, ooh, that's like, I don't know what it's a big deal. So it gets out to a really wide audience and gets more people thinking about PWS. And, this program is very broad, and so the publications have been everything from understanding the basic biology of PWS to developing new models to doing clinical trials. The diazoxide trial, for example, was supported through this program to get it started. And we see a great return on investment. So again, a lot of what we do is start projects that we can then, people can get funding for. So people who have started their projects under FPWR have gone on to get a lot of money from government agencies to continue those projects. It's helped develop new cells, the SNORD-116 animal model that many investigators are using. That was one of our first projects that we supported probably in 2006. And it's being used across the globe now. So new drug targets, new potential insights into clinical care and clinical trials. And I just show here just some stats on it. This graph shows that we kind of evenly split between in vitro, so sort of just in a lab dish, studies in animal models, and then clinical studies that are either on individuals with PWS or clinical trials. So it's kind of split up fairly evenly. This is the number of grants we have supported through the years. We're not done with 2017 yet. Now we have some, our budget is split now between the directed and the grants program. And this just shows you the breadth of this program, that we have grants in behavior, endocrinology, genetics, muscle, neurobiology, so across the board. And we keep track of the results that come out of here. Here are some of the papers that were published so far this year, just a few examples. And you can see, obviously, you don't want to read all this, but what it shows is that it's everything from brain imaging to molecular processing to autism. So all kinds of things are being generated through this program. And I encourage you to try to stay up to date and see what your funds are going towards. We try to communicate that to you in many different ways. The scientific abstracts will be up on the website this week, so if you want to breeze through those in your late nights, you're welcome to do that. We have webinars on our new grants. We'll be having new grants coming out in the next probably month to 2 months that we can talk about. So please sign up for our webinars. We're trying to keep them a little bit briefer so we can just give you a few punches of what's going on. And then on our webpage, you can always go to the research outcomes, where we list all of the outcomes from our research projects. And under each project, we try to keep up to date the things that have come out of those projects. So if you ever think, what happened to that project, blah, 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 you can go on the website and see what the papers and things that have come out of that. So that's our grant program. I'll mention one of our new genetics efforts, because I think it's really cool, and that is our PWS Genome Project that we hope to launch at the end of this year. So, and this is a part of our directed research program looking at genetics and what we call genotype to phenotype. So how does the loss of the genes cause the symptoms that we see? What's that link? So this program is really, all of the projects in this program are directed towards that question, and this is one of the projects. So we know that there are a lot of clinical symptoms in PWS, so hyperphagia, growth hormone deficiency. Almost all of our kids have growth hormone deficiency, but Daniel has hypothyroidism, and most kids don't have hypothyroidism. And some kids have seizures, but it's a small percent of kids. It's about 20-25%. And some kids have scoliosis, but not everybody has scoliosis. So what are the things that are making my son have hypothyroidism and your child have seizures? And how can are there better ways to predict that, and so that we can monitor that? So, we know the PWS region is on chromosome 15, right there. But you'll look, that's a very small part of all of our chromosomes. So there's 20,000 genes. So we talk about like the 4 genes, well, it's really more than 4 genes in the region. But there's 20,000 genes in our genome. And, for all of us who don't have PWS, we have gene variants that are influencing our susceptibility to obesity and our susceptibility to seizures and all of these things. So it's logical that those genes are also, all those other genes are impacting our kids. And those genes also are important in how we metabolize drugs and how our safety profile of drugs and how drugs work, because different drugs work differently in different people. We know that already, and the same is true in our kids. So can we gain some knowledge about that and use it in the care of our kids, and also to predict which kids are going to do well on what drugs? So with the Genome Project, our goal is to sequence PWS genomes and look at those DNA variants, and then use that information to better understand the basis of the variability in PWS symptoms. So we know there's environmental influence, right? But there's also genetic influence. So what is the genetic influence on how people vary in their symptoms? better understand the risk of those symptoms that are not 100% in PWS. So can we say, this child is at higher risk for developing scoliosis, and this child is at higher risk for seizures, so maybe they need to be monitored slightly differently. Full disclosure, I'm not sure that we're going to be able to do that, but the only way we're going to be able to figure out if we can do that with good efficiency is to do the project and see. And then also to predict who's going to benefit from drugs and inform drug dosing. So let's take the example of oxytocin, right? So we already know from the clinical trials, there's some kids who, at least in the studies that have been done to date, some kids who seem to have a big reaction to it and some kids who don't seem to have as robust a response to it. So that might be because there's known variants, for example, in the receptor to oxytocin that influence how sensitive a person is. So can we look at our kids, if we had that information ahead of time, can we put them into clinical trials differently based on whether they have a sensitive or less sensitive? So can it inform who gets what dosing? And can that make our clinical trials better at quickly seeing what the dosing should be, for example? And safety, too. I mean, we know with the story with the beloranib, which was a drug that really seemed to help the hyperphagia in PWS, but there was a problem with blood clotting. Well, if we knew about blood clotting is one of those things that's very variable between people and has genetic influence that's well defined, if we had that information, would we have been able to see whether someone was at more or less susceptible to that? So moving forward, sort of monitoring drug safety in the long term, having that genetic information. So our goal is to, we're in the development stage now. There's a lot of different technologies. Genetic technology is really advancing quickly. So which approach are we going to use? There's whole exome sequencing, there's whole genome sequencing, there's these chips that have all of the known DNA variants on them. So we're sorting through how best to use which technology. Data security, it's a tremendous amount of data. There's terabytes of data per person, so just storing that in a secure place is really important. And then there's ethical issues that go along with that. So scenario, right, you sequence your child's DNA and you found out that your child has a gene for early onset Alzheimer's. Well, that means you or your husband's carrying a gene for early onset Alzheimer's, right? So those ethical issues of these unintended consequences when you sequence a person and how you convey that information back to families, because sometimes it's a real strong, well-known association, and sometimes it's just that it looks like it could be, and so there are a lot of ethical issues that we want to get the community's input on handling. But our goal is to do this DNA sequence analysis to detect the variants that we know already to impact the risk, may impact risk and severity and drug safety, and link that information to the data in the global registry so we can look at what complications your child has and how that relates to their DNA sequence. So, and give that information back to you so that you can use that information in working with, for example, a psychiatrist on drug selection for psychiatric drugs. So, so all of that. So we're planning on launching this at the end of the year and into 2018. Anyone who wants to be enrolled will need to have all of their data up to date in the global registry, since that's going to link. So hopefully a little bit more motivation to get you into the global registry and get your surveys done for that. And I'll just close my section just by mentioning that, one of our goals in FPWR, as Natalie mentioned, is, we learn from the entire rare disease and drug development community. So it's important for us to be very actively engaged in that community, and we really are. So I'll just throw out a couple of examples. Natalie gave the example of the Preclinical Animal Network being featured in BioCentury, which is a trade journal. I'm a member of a collaborative group, the FDA, Duke University, and several academic groups, and then many industry partners, the Clinical Trials Transformation Initiative, and that's CITI. CITI works on important challenges in all clinical trials and improving the clinical trial process in the U.S. And I've been involved, for example, in using mobile technology to develop endpoints, and the FDA is highly involved in that because they want us to use more mobile clinical things in clinical trials. You know, use your phone, use your apps, and how do we do that in a way that is acceptable to the FDA. So I've been involved in that project just as a team member, and it was a great experience. I'll be presenting that at the NORD meeting, the National Organization for Rare Disorders, in October. Our organization is very involved with NORD. Jessica sits on the registry advisory board there. Susan was invited to join the board of directors there. I'll be presenting at their meeting, and Jessica will be presenting at their meeting. She'll be presenting our grant that we got from the government agency, PCORI, on using mobile technology in PWS. I was invited to the National Academy of Sciences, actually, a couple of months ago to talk about our PWS Genomes Project, because they have a workshop on using precision medicine in clinical care. And we have fairly frequent interactions with people at the NIH, the National Center for Accelerating Translational Sciences, and the NIH, the overview of the big NIH, as well as the FDA. So we're really constantly involved in all of these things. And I mean, I feel like the more we get PWS out there and the more people see PWS as sort of a model disease to be doing drug development in, the better it is for our community. So that's my part, and if we have any questions, I'd be happy to address them.
So Arthur had the three testing. He had the FISH, the methylation, and a microarray. Is that considered all the testing we need to know for the DNA sequencing, or?
So that's just of the PWS region, and it tells you if he has PWS or not.
Yeah.
So this is a project that looks at all the other genes. So the answer is no, he hasn't had all of the other genes.
How would we go about to get that done if we wanted to just kind of be able to put it into
you
the
would
registry?
enroll in the PWS Genome Project, which is going to launch at the end of the year. So FPWR is going to support that. So what we would do is we would make sure that you're in the registry and that all your surveys are up to date, and then we're going to send out kits for you to swab the cheeks of your children, and then you're going to send that back in, and we're going to do the DNA analysis and provide information back to you about that.
Okay, thanks.
Okay, so I'm not going to pretend like I know what's going on. But like one of the things that's kind of a Facebook craze right now, not just in the PWS community, is the MTHFR mutation. And I'm just wondering if there's been any thought or any type of study looking into how often that may occur in our kids and how that may kind of affect the way they metabolize certain things and maybe help weed out, you know, and kind of show what drugs they may be more susceptible to have a good response to.
So again, the Genome Project is going to look at all those genes. So that's the kind of information that we can, if we have all of the information in the registry, what drugs your kids have been on, did they work, and we also know what variant they are, because that's one of the genes. I mean, it'll look at all of the genes. And you know, these you know, it's all those genes too. All of those genes, we're going to have the information. That's why it's terabytes of data. And you can pull those genes out and say, do people with, you know, mutation in that do better or worse on these drugs? So that's why we're really interested in gathering all that data, because then you'll come to us and you'll say, well, what about this mutation? And we'll be able to run it all and say, it doesn't seem to have an impact, or it does seem to have an impact. And, you know, so yeah, it'll be part of that.
Awesome.
So the Genome Project, will you all cap that enrollment, or is it something that you're gonna?
So we're gonna pilot it. So the different technologies cost different amounts, and so that's what we're working through now. So we're looking at piloting 100 people to start, and then we're gonna, you know, run it through and see about the Data analysis, of course, is the biggest thing, right? You get 3 billion base pairs of information, and then, sorting through it is the issue. So I think we're going to try to pilot it on 100 individuals and see how that goes. But yeah, I mean, I think we would, the goal would be to expand it to everybody who's in the registry and have it as, your DNA sequence isn't going to change over time. So once you have that information, you have it for life.
Right.
So, then we can build questions. If new drugs come along and some people seem to respond well and some people not respond well, we can build those questions right into the existing data.
Great, thank you.
Is there an example of another rare disorder, or who else is doing this?
So, what I learned at the National Academy, actually, because it was a workshop and it was all the big pharmaceutical companies, all of the big pharmaceutical companies that are doing cancer trials and, heart disease trials, they take DNA on every patient that enrolls in their trials. They do a genome analysis because the cost has come down. So the first genome was sequenced in 2003, and it took $1 billion and cost 10 years, and now you can do a genome for $2,000, right? They get all that information and they keep it because they want to know who responded and who didn't. But they don't have a mechanism to give that information back to anyone, so that the patients never know. I mean, they know that it's being done. They don't do it in secret. I'm not saying that. But the companies don't relay that information back because they're not genetic counselors. They're not, they don't have, what I heard from the companies was, we'd love to share that information, but we're not going to hire a team to share that information with the 3,000 people that are in our clinical trial. So it's actually being done all the time now. So
it's kind of like a super duper advanced 23andMe.
Yes, exactly. That's exactly what it is. Yeah.
So I know some other people that have had a more challenging time getting a diagnosis have already had a whole exome sequence. Is that something we're looking to filter in? Is that, there might be people within our population that have already had the West test completed? Is that information that we can take and apply to the study? It
would depend on where that information is and how it's stored, and usually if it's your information, you can get access to it and share it. But so that's actually a great idea.
Okay.
Yeah.
Thanks.
One more.
Just kind of completely random thought, but are there any synergies that could be made working with Angelman's? Do they have a similar organization like this, knowing that it's the same genes that are affected just in a different way? Are they trying to do the same thing, maybe from a gene editing standpoint or things like that, where that could be beneficial to?
So actually, they're doing the opposite. So we're trying to turn the genes on. They're trying to turn our genes off. And actually, there's a lot of synergy there, right? Because they need the same thing. They need to get into that same region. So many of the scientists that are working on PWS also work on Angelman. So you saw Larry Ryder yesterday, Stormy Chamberlain, who is here at the conference, and Dr. Jang, actually, who's done the gene activation in the mouse model. They all work on Angelman as well. So actually, at a scientific level, we're working with a lot of researchers who, because that area is so complex that those people have an understanding of kind of the flip-flop between the genes. So we don't have any formal association with the Angelman Syndrome Foundation, although we've gone to their meetings and know the people. So yes, it's definitely an area that we could probably forge better. We could probably continue to build those relationships, but there are a lot of connections that we're trying to capitalize on. All right. Should we? Oh, sorry.
I have sort of two questions pulling back to the very beginning and everything we've talked about today. One is, what do you guys see the biggest challenge in terms of resources? I've heard you talk about seeking talent, seeking different types of experts who are interested in this to devote their time and energy toward it. There's obviously money as a big component, and some of it is just letting these things run their course and taking the time to do the science. But if you made a list of the things that are the hardest about this challenge, is it money? Is it interest from industry? Is it attention from experts? I'm curious about that. And then also, sort of separately, I think for non-scientific people, whenever we hear about CRISPR and genetic research, everything that you read about it that's not PWS related, there's always sort of but what are the ethics? And I, as a person who's not interested in a designer baby, as a mother of a very real child, I just wonder how much of those ethical discussions are about things. How do the ethical discussions split between things that don't exist and helping people who do exist?
So I would say that ethical questions, I think there's very little resistance to using CRISPR technology to address diseases in the scientific community, in the FDA. I think the FDA is very open to that. If it's safe, I think those therapies will move forward. It's a newer technology, so they want to make sure it's safe. If you're editing the genome here, they want to make sure you're not editing in other places. So I don't think there's a whole lot of, it's not really different than gene therapy in that, the ethics around gene therapy. And, the FDA and the academic community is very comfortable with gene therapy for known diseases. Where it gets into the ethical issue is if you're saying, okay, now we're gonna make someone smarter or have better hair or whatever, and that's where the ethical debate is. So I don't think that's an issue really for us. To step back, what are the challenges? Oh, that's a huge question. So I would say money, of course, the more, and it's because of all of you that it's really time. So because of all of you, the foundation has gotten enough money that we can, have a scientific research team, and you see all of us are doing different things. So I think that, it's that, it is time, but time and money are pretty linked, right? So with more money, we can get more people, and we can support more scientists, and we can get more things done. So,
maybe I can add to that one thing that is particular to the biomedical research is very fragmented. So if you remember drug discovery, preclinical animal network, clinical trials, all this require different expertise, as well as there are different organizations that are involved in these different steps. And it's getting better, but everybody has his own agenda. Usually people don't talk to each other, not because they don't want to talk to each other, but it's very fragmented. And this is why also our role as a foundation is really, gluing all these people together. And sometimes it can be, as easy when some people ask me, what is translational research? Sometimes I said it's just putting people with different expertise coming from different type of organizations that sit together around the table, But that's, our role is really that, and that's one of the also big challenges, is really trying to glue all these different steps together, in addition, to what has been discussed as challenges.
Well, thank you for everything you do.
I have